identical genes, one can maintain its original function, the other one can now evolve to give me a new property. And so there are, in all species, there are what are known as gene families. And gene families result from duplications, where you have an original gene, and over evolutionary time, through duplication, there are now multiple versions of that gene, and many of the duplicates diverge in structure and function. Like one of the best known gene families is the globin gene family. The globins are proteins that sort of carry this structure called a heme that helps you to carry oxygen and CO2. And there are different versions of these globin proteins. There's alpha globin, there's beta globin, there's fetal globins. Okay? And fetal globins are very important. You think about when baby's inside the placenta is where maternal blood and baby blood, fetal blood, get real close to each other so the baby can extract oxygen into its blood from the mommy. So it needs a hemoglobin that's got a stronger affinity for oxygen to pull away from mommy's blood. And fetal globin has a stronger affinity for oxygen than adult hemoglobin. Does that make sense? So it can pull the oxygen out of mommy's blood into the fetal blood. And then once you're born, that fetal globin is no longer expressed. You get the mature globins, alpha and betas. So these different genes, through duplication, produces a range of proteins that add functionality. In this case, the globin gene family producing a fetal globin that allows the fetus to extract oxygen more effectively from mother's blood than it would without it. Okay? So duplications can lead to long-term increase in variation in the species. Translocations, here's an inversion. Well, translocations. The translocation is when a part of a chromosome is transferred to another chromosome. Okay? Here we see, and sometimes it can just be a piece broken off and then incorrectly stitched onto another chromosome. In some cases, two chromosomes break, and there are double repair mechanisms. Sometimes the repair mechanisms stick the broken pieces on the wrong chromosome, and we call that a reciprocal translocation. Okay, right? And some of them can lead to serious issues. Now you think, okay, I got a translocation, I still got all the genes there, it just got moved to another chromosome. What's the big deal? Well, it is a big deal because genes, they have to be controlled in terms of when they're activated or not. The controlling elements are often near the gene. So if you have a controlling element for a gene that says when this gene is on and off, and the gene that it controls now gets moved somewhere else, it's now under a different control system. And if you've got a cell if you have a gene that's controlling cell division, and it gets moved to another part of the chromosome or another chromosome that's now under the control that says always stay on, now the cell's dividing out of control and you have cancer. And there's an example of that in terms of a translocation. So you can see the example here between these two chromosomes, two non-homologous chromosomes, where this piece got switched to this piece. Okay, so FGH, this segment got transferred to the KLM here, the Dutch Airlines, and N went to this guy. So it's a reciprocal translocation. Okay, I'm going to go to the next slide and come back to this. Here's a famous classic example of a reciprocal translocation that leads to chronic myelogenous leukemia, a very serious type of blood cancer. This was first detected in Philadelphia, so this is known as the Philadelphia chromosome, as you see here. There was a reciprocal translocation that means DNA chromosomes, pieces were broken and traded between two different chromosomes, the translocation between chromosome 9 and 22. And what happened, it brought two genes together that shouldn't be. And ABL is a gene that influences cell division. And now ABL next to a new neighbor, this gene is on continually when it shouldn't be, and it drives cells that have this translocation into repeated cell division to cancer. Okay, and this translocation tends to occur in bone marrow. It leads to, again, leukemia is an overproduction of white blood cells at the expense of red blood cells, so you're quite anemic, and under severe conditions, it's a lethal disease, or lethal condition because of this translocation. Okay? Go back to inversions. Sometimes a chromosome, part of a segment, is inverted. So here we have EFG, here we have GFE. Okay? again, all the genes are there. You might say, well, that doesn't look too serious, but again, position of a gene, because you think of a gene as maybe a sequence of nucleotides that codes for a protein, but that gene has to have a control mechanism to turn the gene on and off at the appropriate time. And if it gets moved and now it's no longer near its appropriate controlling portion of the chromosome, then its control might be disrupted, leading to serious conditions. Okay. Now, what I'm going to show you now is I think it's a 4 to 5 minute clip. I waited now to do this to show you. You have a question?
What are the control mechanisms again?
Well, I haven't really talked about those. That's the chapter coming up on the control of gene expression.
Okay.
But when you think about when I say gene expression, let me make sure that you know what I mean here. I think you do. But when I say gene expression, I mean that a particular gene encoded in the DNA nucleotide sequence. When we need that gene to produce this product, it's transcribed to messenger RNA, which is then sort of as a working copy of the gene that the ribosome can use to make a protein. There's our gene product, and this is, again, the functioning hardware of the cell. Here's the information for that cell. Now, I think I told you in the last lecture that 30 to 60% of all genes in a cell are on. The rest are off, except for the housekeeping genes that are always on because they have to maintain the critical life functioning of a cell. So different cells, different tissues use different genes. I mean, every cell in your body has genes for the globin family, okay, to make the globin proteins for hemoglobin. But those are only going to be expressed in bone marrow cells who are making red blood cells. Those genes are never used in skin cells or anything else, okay? So there's some control mechanism that says in certain tissues I'm going to turn those genes on, in other ones I need to turn it off, okay? Now, if we look at I'm going to jump the gun here, but if you look at a stretch of a chromosome, maybe here we've got a sequence of nucleotides. Maybe this is the protein coding portion of a gene, okay? All right? And usually upstream of that, there's a piece of the DNA called a promoter, okay? And this is where the machinery binds to turn this gene into RNA, okay? An enzyme called RNA polymerase will bind here and make an RNA message. Now, that enzyme has to bind here only when it's appropriate. And at other positions along the chromosome, sometimes upstream, sometimes downstream, there are other places where other proteins can bind that can enable this enzyme to bind here or not, or block the site. So around a gene, there are parts of the DNA that have sequences that are essentially controlling elements to turn this gene on or off. That's what I mean by that. So you can imagine if a gene gets moved and it leaves its control system behind, it may lead to overexpression, where this is always on. If that gene stimulates cell division, you're screwed. You're going to run away cell division, right? Or it may turn off a critical gene, okay? That maybe this gene is involved in DNA repair. Maybe it senses mutations and fixes them. Maybe if this gets mutated and now it gets to a controlling unit keeps it off, you've lost part of your repair mechanism. Now you're screwed, right? So position of a gene is critical. So all these changes in the chromosomal structure can lead to sometimes serious issues, but not a guarantee, because remember I told you most of the DNA in a human is non-coding. So a lot of these changes, sometimes they're kind of neutral, okay? All right, so I'm going to show you this clip from the Howard Hughes Medical Institute. It talks about the evolution of the Y chromosome, and I'm doing it now because a lot of these events have contributed to the shrinkage of the Y chromosome and its evolution from an autosome to a sex, the male determining sex chromosome. Let's see if I still got that up here. This is from the Howard Hughes Medical Institute. It's a lecture I found on YouTube. I don't know who the professor is, but I don't see sound, so hold up. Let me start over with this. Sound work here.
So we're going to focus on a set of chromosomes coming down here, and this set of chromosomes is eventually going to make it all the way down to man. And as it comes down here, we'll blow it up. We're going to focus in particular on this one pair, an ordinary pair of autosomes that will become the X and the Y. We'll call it proto-X and proto-Y. Now we know that in meiosis, things first get duplicated So we're going to the 4n stage. And this is just a reminder of what you saw yesterday, that this
Crossover.
ordinary pair of autosomes destined to become X and Y can recombine and shuffle genes. They can do it at either end, like so. This is all review for you. This is all familiar stuff. And just to remind you that recombination can occur anywhere along the length of this ordinary pair of autosomes. And just to simplify, throughout the rest of the animation, we'll just show one version of the X and the Y. Here is an ancestral gene which has now mutated on the proto-Y. This is the initiating event in sex chromosome evolution. We got this tyrannical male determining gene. And here is RPS4, again, an ancient ancestor which survives today on the Y chromosome. Now, how was recombination suppressed between the X and the Y? Well, it turns out the Y underwent a series of inversions, just like the one shown here. You'll see a few more. And as a result of the inversion, the X and the Y can no longer nicely align. And if they can't swap parts, what's going to happen? The fruits and veggies on the Y rot. The ultimate form of rotting is just plain getting lost and deleted. So the Y is going to get shorter. And now recombination is restricted to these regions that are still nicely aligned, nicely paired out at the ends. All right, we're going to zoom back to our timeline. We're now going to cross the point where we part company with the duck-billed platypus. And we're going to have some of the same themes. We're going to have some of the same themes reappear. We're going to have this Y undergo an inversion, which is a sort of an internal recombination event. It's